Hello, my name is Leon Palomera. I am studying the second semester in the Laboratory of Molecular and Cellular Biology under Professor Chutongkyu. Today I will talk about this paper that was published in the journal Nature in February of this year. It is titled Two Conserved Epigenetic Regulators Prevent Healthy Aging. I chose this paper because in our lab we're studying neurodegenerative diseases like Alzheimer and Parkinson's disease, and this paper sheds light into new mechanisms involved in healthy aging. The corresponding authors of this paper are Shiqing Cai from the Shanghai Research Center for Brain Science and Brain-Inspired Intelligence and Dr. Lubin Yang from the Institut Pasteur of Shanghai Chinese Academy of Sciences. These both professors' groups are interested in elucidating molecular mechanisms and biological consequences of the epigenetic factors and how can they affect different biological processes including aging. The lifespan of an organism comprises both the growth and the aging stage. Growth ends with sexual maturity, and aging is characterized by functional decline and increased risk of disease and death. It is important to note that the lifespan does not always correlate with the health span, which is a period of relatively disease free condition. That is why finding the mechanism involved can help scientists develop a way to better understand and even modify the aging process. This paper focuses in two epigenetic regulators that contribute to healthy aging. These are the BAS2 and SET6. The epigenetic modifications refer to the heritable genomic regulation processes that are not directly encoded in the DNA. The most common actors in the epigenetic regulation are referred to as writers, which introduce genetic modifications, erasers, which remove these chemical markers, and readers that are in charge of interpreting these modifications. In this article, the authors found that the BAS, which is a neuronal epigenetic reader, and the SET6, which has a methyltransferase activity, accelerate behavioral deterioration in C. elegans and also in the mice model and in two cell lines. Scientists have tried to understand the genetic regulation of aging since the discovery that some invertebrates that show mutations that allow them to live significantly longer lives than their wild-type counterparts. Several of these modifications have already been found. Some of these modifications can be used as biomarkers of age. That is the case of DNA methylation, which is an accurate predictor of both chronological and biological age. Measured in the chronological age is defined as the age of an individual measured in time units, and the biological age is the years of the population average that is most similar to the individual condition. That is why DNA methylation has been used as a marker of different states of health during different periods of time. The differential expression of the epigenetic regulators described in this journal make the difference between healthy, regular, and unhealthy individuals of the C. elegans species at different time points. This table, extracted from a review paper published in 2015, shows previous findings regarding the histone 3 lysine 9 trimethylation in the aging process of different species like Homo sapiens, Melanogaster, and C. elegans. One challenge found in the past is that the different effect caused by the same modification when is present in different cell types or different species. Another challenge about determining the effect of the epigenetic modifications is that these changes can come as a result of different stimuli. Even lifespans among isogenic organisms is highly variable under control conditions. Some of the most important agents of epigenetic regulation are shown in this figure. We can see that, for example, diet or dietary restriction has mostly an increased effect on lifespan. Differences in how animals react to the circadian rhythm or circadian cycles determine if their lifespan are going to be increased or decreased. Things like exercise, pheromones, or even exposure to sex steroid hormones can also affect the lifespan of an organism. The authors of this paper needed to find a reliable method to determine the outcome of the epigenetic modifications that they were studying in their models. That is why they based their research in this paper published by Jing and colleagues in 2014. They demonstrated that changes in the level of neurotransmitters serotonin and dopamine are associated with age-related behavioral deterioration. They found that a reduced expression of the dopa decarboxylase base 1, which is in charge of producing serotonin and dopamine, was decreased as time passed, and it impaired pharyngeal pumping, food slowing responses, and the male mating in the C. elegans model. This dopa decarboxylase BAS1 biogenic amine synthesis related protein 1 is conserved in humans and is responsible of catecholamine biosynthesis and serotonin and melatonin biosynthesis. The other two proteins, the epigenetic reader BAS2 and the methyltransferase SET6, also have human orthologs. The human orthologs are EHMT1 and BAZB2, and we can find in the protein atlas that they have already been characterized as playing a role in transcription regulation. This figure shows the similarities between the original C. elegans protein and the human orthologs. It shows the similarities in the amino acids and domains. And in the figure B, all the in black are marked all the similarities between also the human orthologs and mouse orthologs and the C. elegans original protein in different domains. After analyzing the results from previous publications, they decided that they were going to use the BAS1 expression as a marker of healthy aging. So they performed first an RNAi screening in C. elegans using the ARINGEN library in mutant worms expressing TU3401, which enhances dsRNA uptake by neurons in the C. elegans, and they also expressed a green fluorescent protein attached to the BAS dopa decarboxylase. After that, they performed C. elegans behavioral assays, and then they validated the results obtained in a strain of mice expressing a knockout mutation for the BAS2B ortholog. And they also used two mammalian cell lines, one from mouse neurons, primary neurons, and one model of HEK293T human cells. All the levels of the dopamine and serotonin were measured by HPLC. The C. elegans behavioral assays that they performed consisted of pharyngeal pumping quantification, which is just counting how many pumps they can perform in 10 seconds. Male mating, which consists of the emergence of more than three male progeny. If we remember, C. elegans has two different sexes. They can be hermaphrodites when there are no males, and also males that can mate the hermaphrodites and produce male progeny. Usually, C. elegans in harsh environments only reproduce through the hermaphrodite way. Basal slowing response and enhanced slowing response account for the tendency of worms to slow their movements when they are in presence of food. The basal slowing response quantifies the body bends that the worm makes in 20 seconds. And to measure the enhanced slowing response, what they did was deprive the worms for 30 minutes after the basal slowing response test. The stress assays that they performed were a hydrogen peroxide test in which they were added different amounts and concentrations of hydrogen peroxide, and after 4 hours they added catalase to inhibit the stress response from hydrogen peroxide, and they evaluated mortality. The other stress assay that they performed was exposing the worms to 1,600 joules per square meter of UV light and assessing their mortality. And also, they exposed them to 35 degrees Celsius of heat and assessed their mortality every 2 hours. For the mouse model validation, they did primary neuron culture of mice in the embryonic day 14 and postnatal day 6. And they used electroporation to add a lentiviral infection of the BAS2B and EHMT1 methyltransferase to use these neurons in vitro. And they also performed mouse behavioral assays in the mutant mice expressing BAS2B knockout phenotype. They performed the open field test, which they evaluated through a video tracking system. They performed the Barnes maze test, which consisted of a session of training and then a session of testing the ability of the mice to recognize the escaping hole. They also performed a novel location recognition, in which the training session there was a mouse exposed to a closed space with two different objects, and the next day they moved one object to a different position and evaluated the time that the mice spent exploring these two objects. And they also performed a lifespan assay and determined that the end of the mice days were when mice were not eating or drinking for 24 hours or lost 20% of their weight. So the first part of the experiment consisted in recording the basal expression of PAS1 decarboxylase in different stages of the life of the worms. So this figure A shows a fluorescence of PAS1 in the neurons of the worms in day 1, day 7, and day 11, and we can notice that the relative fluorescence decreased as the days went by in the life of the worm. Figure B shows us the way they performed the genome-wide RNAi screening. They used the same GFP expressing PAS1 worms. So they introduced RNA interference to knock out around 80% of the genes of C. elegans. They performed 3 rounds. The way they did it was they scored these clones, they divided it into low GFP level as negative clones and high GFP level as positive clones. So high GFP level clones passed to the next round. So in the first round, they collected 520 clones, then 126 clones, and in the end, they found 59 clones. That means 59 genes that, when knocked out, increased the expression of BAS1 in the adulthood of the worms. And these 59 clones included, among other genes, the SET6 and BAS2 genes. Figure C shows us the relative mRNA level of the BAS2 and SET6 and how they increase as worms age. For figure D, they already used mutant worms expressing a knockout mutation for BAS2, SET6, and a double knockout mutation for BAS2 and SET6. And they compared the expression or the levels of both dopamine and serotonin at day 1 in white and day 9 in gray. So we can notice that compared to the wild type expression or level of these proteins, those worms that show the BAS and SET mutation show a higher level of this protein, which means a healthier state. It is important to note, though, that the effect is not additive. It is basically the same level with just one mutation or two mutations. This figure shows us the information that they used from the results to select BAS2 and SET6 to validate this model in the following experiments. So in the figure A, they show us the fluorescence at day 2 and day 9 in 4 different groups using the empty vector as a negative control and the E2 knockout mutation as a positive control. It has been previously reported that the E2 preserved the state of the neurons through an increased expression of dopamine and serotonin compared to wild type worms. We can see in those images that the fluorescence is very much increased compared to wild type when worms express the BAS and SET6 mutation. Figure B shows us how in the x-axis, in the y-axis, the relative BAS1 GFP level, and in the x-axis, these are different genes and the negative logarithm of the p-value. And we can notice that BAS2 have a high p-value and also a high relative expression. And that is why they decided to choose these two genes. Figure E shows us the pumping rate in times per minute of different gene modifications that they found during the screening in day 1 and day 7 of adulthood. And the BAS2 and SET6 show a pretty high pumping rate compared to the other genes. So to test these previous results, they confirmed the basal expression of these two proteins, BAS2 and SET6. They performed a Western blot analysis at different time points, and then they confirmed that the expression levels were time dependent. We can see here in this Western blot image the increasing concentration of BAS2 and SET6 in day 5 and day 9. So both these proteins have a tendency to increase as worms age. And here we can see the preserved fluorescence of BAS1 in SET6 and BAS2 mutants, and how the relative fluorescence at day 10 is very similar to day 2 in the mutation groups compared to the wild type. So for the mice model, they had to generate a BAS2B mutated wild type on a C57BL6 background. They used small guidance RNA and Cas9 modification to create these mice, and figure A shows us how they did it and the confirmation through Western blot, how these heterozygote and homozygote knockout mice show different expression of BAS2, and they use tubulin as a positive marker. Figure C shows us the oxygen consumption rate of mouse microglia, mouse mitochondria in different areas of the mice brain. Uh, Only the BAS2B knockout showed a significantly increased oxygen consumption rate compared to the other mutations. Figure D shows us the results of the open field test in 3 different groups: the wild type group, the heterozygote BAS2B knockout, and the BAS2B homozygote knockout. But there are no significant differences in these 3 groups. The results of the behavioral tests in C. elegans show that deletion of BAS2 or SET6 delays age-related behavioral decline and also extends lifespan. Figure A shows the pharyngeal pumping rates of C. elegans in different groups, showing a statistically significant increase in the pumping rate in the BAS and SET mutation groups. Figure B shows us the mating efficiency of the mutated groups compared to the N2 wild type, and we can see that the mating efficiency is increased in most groups compared to wild type. Figure C shows us the basal slowing rate counted as body bands every 20 seconds. We can see that the young worms have a similar reduction in the movement rate in all groups, but the aged worms show a significantly more evident reduction in movement in most of the mutation groups compared to wild type, which means a better response. Figure D shows us the enhanced slowing response in young and aged worms in all of these groups, mutated groups, compared to wild type. And it also shows that compared to wild type, there is a better enhanced slowing response in the mutated mice, especially in SET6 and BAS2 mutations. And in figure E, we can see the survival percentage, and we can note that it is significantly different. That means that mice expressing the BAS and SET mutation lived significantly longer than the N2 wild type worms. This figure shows us the results of the stress assay, and we can note that same tendency happened here. Mutated worms survived better to hydrogen peroxide at the time of the experiment and also as days went by. It is important to note in D, E, and F that they compared the BAS and SET mutation with the DAF16 mutation, which is insulin pathway mutation E2 that causes dietary restriction in response to the mutation, and also CLK1, which causes reduced mitochondrial function. So it is important to notice that even the mice, the effect found from the benefit found from the BAS2 and SET6 mutation is impaired when it is combined with CLK1 mutation, meaning that the BAS2 and SET6 work through mitochondrial function. So to confirm these results that BAS2 and SET6 regulate uh, the histone 3 lysine 9 methylation, they performed an assay to find out if they were producing monomethylation, dimethylation, or trimethylation, and they found in the results that the most prominent effect of the SET6 methyltransferase causes a trimethylation. This number B shows an overlapping of the numbers of genes that are bound both by BAS2 and SET6 in a immunoprecipitation sequencing data. And here in number 6, it shows a clear interaction of the MRPL49 and MRPS7, which are mitochondrial function genes, interacting with BAS2 and SET6. When they performed a gene ontology in BAS2 and SET6, they found that they are involved in ribosomal protein and mitochondrion protein. So, taking this into consideration, they performed a ATP and oxygen consumption rate in BAS2, SET6, double mutation, and also overexpression. And they found that overexpression of the BAS2 SET6 reduces ATP and reduces oxygen consumption even more than the basal condition. And figure H shows us a ratio of nuclear DNA and mitochondrial DNA, showing us that the effect found from the BAS and SET6 mutation is not dependent of the number of mitochondria. So once they found that the number of mitochondria was not involved, they thought that it may act through the UPR. So this fluorescence shows that the attenuation of the UPR by downregulating the UBL5 and ATFS1 genes prevented the lifespan extension, meaning that the BAS and SET act through the UPR, which promotes mitochondrial proteostasis. So finally, they demonstrated that the BAS and SET functions are conserved in mice and also in human cells. So they found in the literature that the BAS2B expression and EHMT1, which are the human orthologs, are increasingly expressed as the humans age. They also found out in the mutant mice that mice expressing the BAS2B knockout mutation tend to preserve their body weight better and avoid becoming overweight when old. Figure C shows us the quantitative data of the body weight of the mice that is preserved in the BAS2B knockout. So all these results showed that the BAS2 and SET6 contribute to mitochondrial dysfunction through histone 3 lysine 9 trimethylation of mitochondrial upregulating genes. They also found that this mechanism is conserved in mice and also in human cells. They showed that the human ortholog expression correlate with AD progression and aging, and that ablation prevents cognitive decline in mice. Further studies are required to validate these results and maybe propose a therapeutic mechanism involving these methyltransferases to prevent or better the health in aging humans. Thank you.